Hello dear students, welcome to my tutorial channel Brilliant Bios. In this class we are going to learn about protein sorting. This is a very important topic for CSIR-UGC NET Life Science. This is the beginning of this class, so in this class we will learn about some basic facts about protein sorting. So protein sorting means proteins that is produced in a cell must be sorted out. As we all know that cell is a tiny in structure, but a cell contains many different type of membrane bound organelles like nucleus, mitochondria, chloroplast, peroxisomes, endoplasmic reticulum, Golgi bodies, endosome, lysosomes. So there are many different type of compartments or that are called actually organelles are present. They perform different functions. Let's assume an example, then you can understand what is protein sorting. DNA polymerase. This is a protein produced in the cytoplasm, but its destination is into the nucleus. Because in the nucleus there is a DNA, and DNA polymerase helps in the DNA replications. Likewise, RNA polymerase. There are many different proteins are present that are destined to the different locations. So those proteins perform its function in a particular organelles. All the proteins are produced in the cytoplasm, but it must be sorted out. Example like plasma membrane. There are different type of channel proteins are present, transporters are present, our receptors are present. They are also protein in nature. They can be synthesized in the cytoplasm and must go to the plasma membrane. Another example is extracellular matrix proteins. So cells can secrete these proteins. So these proteins are also produced in the cytoplasm but must be secreted out. So in a general, protein sorting means proteins are produced and then these proteins are should be sorted out to its proper destination to perform its function. Like if DNA polymerase is present in the lysosome or in the Golgi bodies, it will not perform its functions because it requires DNA. So at first I will give you two terms to help you understand protein sorting. The first is co-translational translocation. Okay, the second is post-translational translocation. So there is a difference here. Co-translational translocation. Translocation is movement. A protein produced in the cytoplasm, now it should translocate to another region or compartment or organelles. Likewise, in case of post-translational, there is also translocation, but the fact is there is a difference in the synthesis of the protein. When we call co-translational, that means the protein is not produced totally. The protein is producing, it is actually producing. And in case of post-translation and translocation, the proteins are already produced. Okay, I will give you some examples. In case of co-translation translocations, all the proteins that go through co-translation and translocations actually enter into the endoplasmic reticulum at first. You have to understand this. In case of co-translational translocation, all the proteins that actually go through this protein sorting mechanism enter at first to the endoplasmic reticulum. This is specifically rough endoplasmic reticulum. Okay, so protein must enter into the endoplasmic reticulum. Then they go to the Golgi apparatus, Golgi bodies or Golgi apparatus. Then the sorting occurs. Like from the Golgi, the protein can move to the plasma membrane. The protein can go out from the cell to the cell exterior. Example are extracellular matrix protein or albumin or enzymes or hormones, anything. Etc. Plasma membrane proteins like receptor transporter, channels, or ejector. So there are different type of proteins present in the plasma membrane, present in the cell exterior. So these are all proteins that actually called secretory proteins. I will tell you. Then from the Golgi body, the protein can move to the endosome or directly to the lysosome. Okay. So there are 4 destinations for co-translation translocations, where the protein is not totally made. The protein is producing from the ribosome, and it enters simultaneously. It enters into the rough endoplasmic reticulum. After total production of the protein, it enters to the Golgi apparatus. Then it can go to the plasma membrane or cell exterior or endosome or lysosome. This process, this whole process is called secretory pathway. Secretory pathway. This is also known as, very importantly known as, vesicular trafficking. You must remember, vesicular trafficking. Okay, this is also part of protein sorting. Secretory pathway, vesicular trafficking. Why it is called secretory pathway? Because these proteins are actually secreted out. Most of them, like cell exterior proteins, are actually secreted out. They actually secreted. So for that reason, these proteins are called secretory proteins or secretory pathway or vesicular trafficking. Here the protein is not totally produced. But in case of post-translational translocations, the protein are actually totally produced in the cytoplasm. Now these proteins actually go to different location or organelles like endoplasmic reticulum. So you can say here that here is also example of endoplasmic reticulum. Here is also example of endoplasmic reticulum. Okay, the thing is, those proteins that enter through these co-translation and translocations to the endoplasmic reticulum are called secretory proteins. Secretory proteins. Okay, but those proteins that enter through these post-translation translocations are not secretory proteins. They are not secreted out to the plasma membrane, cell exterior, endosome or lysosomes. They are not secreted, or there is no vesicular trafficking occur. What is vesicular trafficking? In my subsequent classes I will discuss. But in case of post-translation translocations, those proteins that enter into the endoplasmic reticulum, they are ER or endoplasmic reticulum resident proteins. So those proteins that enter through this process, they are resident proteins. That means that proteins actually always present in the endoplasmic reticulum and perform its function in the endoplasmic reticulum. So they are called resident protein. These proteins are secreted out in this 4 compartment, but these proteins actually remains in the endoplasmic reticulum. Okay, the next destination is nucleus. Another is mitochondria. Another is chloroplast, peroxisome, etc. Now in this class I will talk about how proteins enter into the endoplasmic reticulum, and my subsequent classes I will talk about how proteins enter into the nucleus, how proteins enter into the mitochondria, chloroplast, peroxisomes, endosomes, lysosomes, plasma membrane, all about this protein sorting. But at first you must remember that the protein is actually entering inside the endoplasmic reticulum for this whole process. So this is very important. So the protein must be destined to the endoplasmic reticulum. So for that reason there should be some features about that protein. So for the secretory proteins, those are secretory proteins, they have some specific you can say specific signature, like we call signal sequence. Signal sequence. Okay. Another thing is, for post-translation translocations, like if the proteins should enter into the nucleus, mitochondria, chloroplast or peroxisomes, they also should have some signal sequence. So signal sequence is very important. This signal sequence, also known as targeting sequence, or you can say signal patch, okay, so, or signal peptide. So the name is very different: signal patch, signal peptides, but they all mean the same thing, that the proteins have some sequence or amino acids that actually help in to reach into these destinations. Okay, the feature about this signal sequence is more or less approximately 20 amino acids actually make this signal sequence. But I am talking about endoplasmic reticulum. So what is the feature of this endoplasmic reticulum signal sequence? Yes, the sequence or the secretory proteins that actually go through this process, they have approximately 16 to 30 amino acids that acts as a signal sequence for endoplasmic reticulum, or you can say rough endoplasmic reticulum. Generally the sequence is approximately 20 amino acids, but those destined to the rough endoplasmic reticulum, specifically the secretory proteins, they have 16 to 30 amino acids. This amino acid sequence, this length, is actually known as signal sequence. And if you want to know more details about this signal sequence, you will learn that there are some positive charged amino acids are present, like lysine, arginine, one or more positive charged amino acids are present, and then there are a stretch of approximately 12 amino acids, 6 to 12 amino acids, hydrophobic amino acids are present, hydrophobic in nature. So there are some positive amino acids, then a stretch of 12 amino acids, hydrophobic amino acids are present. This is called signal sequence for rough endoplasmic reticulum. Those are destined for secretory pathway. This is very important, secretory pathway. Now I have said that this is called co-translation translocations. That means this is a large ribosomal subunit, this is small ribosomal subunits, and the protein is actually producing, totally not produced but producing. Then I am talking about a signal sequence. Yes, there is another information about the signal sequence, is that the signal sequence for this rough endoplasmic reticulum, I am talking about rough endoplasmic reticulum, here is the RER. For RER, this signal sequence, they are basically present in the N terminus. So you can imagine when a protein is synthesized, this is the N terminus. Okay, so in this N terminus there is a signal sequence. I am drawing it in the red color. So this is the signal sequence that is present in the N terminus. Okay, now there is a dedication of a protein that is called signal recognition particle. So there is another information. There is a protein that can recognize these 12 amino acid, hydrophobic amino acids, because for RER there is a 16 to 30 amino acid of stretch of signal sequence are present. These are amino acids, but these 6 to 12 amino acids are very important, because if you mutate artificially this hydrophobic amino acid into charged amino acids, they will not perform its function. The proteins cannot destined to the rough endoplasmic reticulum. So this area, this region is very important. Okay, and this stretch of amino acids can be recognized by a protein called SRP. The full form of SRP is signal. Why it is called signal? Because this can recognize the signal sequence. Signal recognition particle. So there is a protein, let's assume this is a signal recognition particle. They can recognize these signals and can bind. Now there is some information about this signal sequence, SRP, signal recognition particle. That is, the signal recognition particle is not only made up of protein, it is actually ribonucleoprotein. Ribonucleoprotein. What does it mean? Ribonucleoprotein. Ribonucleoprotein means this molecule have protein, also have RNA molecules. This is very interesting. So this SRP proteins have 6 subunits of proteins, and also a RNA approximately 300 nucleotide long RNA are present. So it is called ribonucleoprotein. Okay. Now this is a ribosome producing a secretory protein that has signal recognition particle. I am showing it in red in color. Now SRP protein bind with this recognition particle because SRP has some property by which it can recognize the hydrophobic region of this signal sequence. I am not going into more details about SRP because there is some domain present in the SRP, a subunit called P54. There is a many information about this SRP. I am not going into details, but I am sharing you. There is a subunit called P54. This P54 subunits have an M domain. Why it is called M domain? Because there is a high concentration of methionine amino acids are present, and many hydrophobic amino acids are present. So in this P54 subunit, there are 6 subunits. Among them, P54 is very important because P54 actually bind with this signal sequence, specifically the hydrophobic amino acids. Because this P54 have M domain, this M domain is hydrophobic in nature. So the hydrophobic groove actually interact with this hydrophobic amino acids by hydrophobic interactions. So forget about this. These are SRP molecules. So I am writing here SRP. So these signal recognition particles are present in the cytoplasm. Now it binds with the 12 amino acid hydrophobic region, hydrophobic amino acids. I am calling it signal sequence. Now this SRP will bring this ribosome to the endoplasmic reticulum. You have to understand this. Now this SRP bring this total ribosome along with this protein, or we can say it is nascent proteins it is producing. So it is endoplasmic reticulum membrane. Okay, it will tether this whole complex. So there is 3 things: ribosome, protein, and the SRP. Now in the endoplasmic reticulum, there is a receptor for the SRP called SRP receptor. Let's assume this is a receptor. So it is SRP receptor. SRP receptor. So in the endoplasmic reticulum, the membrane, there is a SRP receptor that can recognize the SRP. Now SRP actually brings this total thing, and not only that, there is a channel-like structure in the endoplasmic membrane. We call it, there is a channel-like structure through which it actually enters. This protein will enter. This is called translocon. So this structure is called translocon. Example, SEC61 complex. Okay, you can remember this. Mainly you must remember this is called translocon. So SRP helps to bring this complex, then this ribosome will sit on this translocon and deliver this protein through this channel called translocon. So this is co-translation translocations. Here there is no requirement of energy because as the proteins are produced, they start to enter through this translocon or SEC61 complex. Okay, now in case of post-translation translocation, there should be some difference. So in this class I have mentioned that I will talk about only the endoplasmic reticulum, not other things. So I have to remove these writings. So I am removing in this part. Now let's talk about the post-translation translocation. What happens when proteins are destined to the endoplasmic reticulum but post-translation translocation? In that case, the proteins are totally produced. The proteins are totally produced in the cytoplasm. Here ribosome are actually producing, but here the protein is totally produced. Now it must go to the endoplasmic reticulum. So let's assume this is the endoplasmic reticulum membrane. In case of post-translation translocation, there is no requirement of SRP. Okay, so there is no requirement of SRP. But the thing is, here also this translocon is present. In this case, there is also this translocon is present. This is a channel-like structure. But there is another thing is also important for post-translation translocation of ear into the ear. This is called another complex. We can say, let's assume this is a protein, this is also a protein. We call it SEC63. We call it SEC63, and we know this is called SEC61 complex. So this is translocon. This is another protein only required for post-translational translocation. What is the function of this protein in the endoplasmic reticulum membrane? Yes, there is a function. As you can see that here the protein is totally produced, so it is called post-translational. After translation, this is translocating into the endoplasmic reticulum. So in this case, the ribosome are producing one by one amino acids, attaching one by one amino acid and producing the protein. So it is the force that is helping the protein to come inside, but there is no force. So here another molecule is responsible for the entering of this protein called BIP. You must remember this. This is the family of HSP70 protein. So what is called HSP? HSP means Hitchcock protein. This is a chaperone protein. What is the function of chaperone proteins? Chaperone proteins helps to keep protein in its linear conformations, not to fold a protein. So when HSP70 like proteins like BIP proteins or Hitchcock proteins or chaperone proteins bind with the protein, it cannot fold. The protein cannot fold. So it actually prevents folding. Okay, now this BIP protein actually carries some ATP. So it is attached with ATP. And this SEC63 actually can break this ATP to ADP. So it can break this ATP into ADP. When this SEC63 protein break this ATP into ADP, the conformation of the BIP changes. Let's assume if this is a BIP, okay, closed in its mouth when it has ATP, but when this ATP is changing into ATP, the structure of this BIP will look like this. It has ADP. Now it has a mouth. Let's assume this is a closed mouth, is an open mouth. When it's opening mouth, protein can actually go through this. It can attach with the protein. It can attach with the protein. Now, then this ATP is actually another time replaced by ADP is replaced by ATP. Then it will shut the mouth. So it will actually helps in the protein not to fold very quickly. So what happens in case of post-translation translocation? The protein is entered through this SEC61 because this is the channel translocon. SEC63 helps to break this ATP to ADP so that the mouth of the BIP can open and can bind with the protein. Now ADP is replaced by ATP so that this will actually become closed mouth. So it will actually tightly bound with the protein and actually acts as a ratchet. It will actually pull down the protein into the endoplasmic reticulum. So this is the beginning of this class, protein sorting, how proteins enter into the endoplasmic reticulum. In my next subsequent classes, I will discuss about in more details about protein sorting. Thank you for watching and always keep learning.